excited we are to have you talking about this very complete. You agree to give a presentation on it. So thank you, Matt. Over to you.
Okay. So, yeah, so there's a lot to cover. I was thinking about making the title of the talk gene by environment interactions, and you know how complicated they are and how I don't actually feel like I understand them. The more I learn, the more confused I become. But I left it with this title. So I think, can everybody, Naomi, can people see my screen? I take that.
I can see your screen.
Yes.
Okay.
Yes, definitely.
Great. Okay. So so I'm going to talk about you know, kind of basically a bunch of kind of issues and interactions that are fairly basic but very important. And I think, you know, I think it's important to go through them even if they're basic, because I do see in the literature often mistakes being made in this regard. And so I even though it's basic, I do think it's important. So I'll cover, you know, two basic types of G by E interactions. So I think part of the issue is that there's confusion about what people mean when they say G by E interaction. I'm then going to talk about scale dependence of interaction, something that a lot of people discuss, but I still think is complicated. It's complicated for me as well. And then models for ways to model G by E and proper interpretation of terms. And I definitely want to get to proper control of covariates. So if it ends up that I'm taking too long, and there's an hour, I think, for this call. So if it ends up that I'm taking too long, then I'll just kind of skip forward to that. And then finally, just a final slide on kind of recommendations for doing G by E research. This is kind of a tip of the iceberg, right? There's a lot of complicated issues in interaction research, but this does, I think, cover the basics of it. So I'll start with the two types of G by E. So obviously G by E refers to the case where the effect of an allele depends on the level of an environmental variable, or equivalently, the effect of the environment depends on you know, what risk, the number of risk alleles that you have. In either case, we'd say that there's a gene-environment interaction. There's two kinds of G by E's if we're talking about polygenic models. And that is interactions where the genetic variance itself changes across the level of E, but the SNPs may be completely in common. They're just affected by a scalar. So can I ask people, can you actually see my mouse when I'm moving it?
Yes.
Okay, thank you. Okay, so you know, this is one way that we could talk about doing this. So if we have a bunch of, you know, K SNPs, you could talk about this as being the sum of, you know, the effect of each SNP times the score at that SNP times 1 plus some interaction term, beta prime E. Well, beta prime is not subset by K, so it's going to be the same across Ks. It's basically just a scalar of the genetic effect. And what this will do, if you look instead of Y at the variance of Y, is the variance of Y will change as a function of E, change across the levels of E. So this is one way that people talk about G by E. In twin studies, this has been done a lot. So for example, there's a famous Turkheimer study from 2003 in which it looks like the additive genetic variance for IQ is lower at low SES and gets higher at high SES. And this is also done a lot with polygenic risk scores. So the R squared, or in other words, the heritability explained from, for example, for a depression-based polygenic risk score increases as a function of childhood trauma. That's a study by Wouter Peyrut. We can also think about a different type of G by E. This is what I call a qualitative. I borrow these terms pretty much from the twin literature, but I think they're helpful. This is where the actual effects themselves are different across different levels of E. And so you can see here in this equation that beta prime K, the interaction effect, is subset by K, right? So the effect actually, there's some, you know, basically what could be thought of as an intercept term, the effect of the allele when the environment's at 0. So that's the plus some effect that's going to change across the different alleles. And what this does is it will, for a polygenic model, will reduce the genetic correlation between individuals of the same genotype at different levels of E. So, you know, there's a lot of examples of this. This is done a lot in candidate gene studies. This is a typical model done when people are looking at individual polymorphisms. So the effect of stressful life events on depression depends on the 5-HTTLPR polymorphism. That's the famous Caspi study, which, by the way, I think everyone knows did not replicate in the very, very large meta-analysis on that. And there's being done a lot with Grimmel methods. So Naomi was on the paper about the genetic correlation between different psychiatric disorders. Also, we did this paper on the genetic correlation of schizophrenia between individuals of African and European descent. You know, if we think about this distinction of quantitative versus qualitative and what types of designs, for twin studies, you know, they're both investigated quite often. So qualitative interactions are looked at, for example, often the question is whether the genes that affect males are the same as those that affect females. That's a qualitative interaction. And the quantitative, the way that I described a second ago with the Turkheimer paper, for example, where the genetic variance changes. For candidate genes, for investigations of individual SNPs or individual polymorphisms, usually what is of interest is the qualitative interaction, whether the effects of the alleles are the same or different across E. It should be noted that, you know, when you're looking at individual polymorphisms, you can't really distinguish between quantitative and qualitative interactions. Even though this is usually the focus, it is the case that if you have a qualitative interaction, it implies that there must be a change in variance across the levels of E as well, even though that's usually not the focus of those investigations. Feel free, by the way, to interrupt with questions as they come. For PRS studies, you know, usually it's the quantitative interaction that's of interest. We're usually interested in whether the R squared is the same across different levels of E. But, you know, you could also look at qualitative interactions with whether, you know, a PRS in one trait or one population of E is correlated with PRS at another trait or another level of E. And finally, for GREML, it's almost always the qualitative interaction that is tested. This is the QG by E or the G by E in GCTA, the software GCTA. That's what's being tested there. It turns out that quantitative interactions are difficult to test with GREML because they require nonlinear constraints. Now that can be done, but you just can't do it with GCTA. You can do it you can write your own. And I think Navard wrote a maximum likelihood script that does that. And we've done the same. So pretty much in this talk, I'm going to be focusing mostly on just single polymorphism G by E, so qualitative G by E. I'll also discuss a little bit at the end G by E for polygenic risk scores, where we're testing whether the R squared differs across levels of E. So scale dependence of interactions. So first, it's important to kind of get our notation correct. So I'm going to call G the genetic polymorphism, E the environmental moderator, and Y the outcome. And for simplicity here, I'm just going to say that they're all binary. So E is 0 if it's low risk and 1 for high risk. And, you know, it's obviously a simplification, so this might be like a dominance model, but 0 for low genetic risk and 1 for high genetic risk, just for simplicity. And we'll call PGE the probability that Y is 1. So we'll call Y the disease, when Y is 1, you have a disease, when Y is 0, you don't, given G and E. So PGE is equal to the probability that your disease given the genotype and the environment. And these are the probabilities down here. So we can talk in this kind of sense, in this way of doing it where everything is binary, of additive or multiplicative interactions. And these generalize to when that's not the case, where everything isn't dichotomous, where, for example, Y is continuous. You can also talk about additive and multiplicative, but I think it's easy to begin this way. So there's two basic ways of thinking about what an interaction means with binary outcomes, and it really depends on how we're modeling the joint effects. So if we're thinking of the additive model, the null for the additive model is that the excess risk for the combination of G and E under the null is just equal to the effect of G plus the effect of E. And so in these probabilities up here, this would be that the effect of the joint effect, probably of having G equal 1, is P11 minus the baseline, right? So P11 minus P00 is going to be equal to the environmental effect, P01, or sorry, the genetic effect, right? P10 minus P00, plus the environmental effect, P01 minus P00, right? So under the additive model, that's the null. Under the multiplicative, the excess risk of the joint is the effect of G here times the effect of E here. So that's going to be the ratio of P11 over P00 over the baseline risk should be equal to, under the null, the ratio of P10 over P00 multiplied by P01 over the effect of E in the multiplicative model. And I should note that almost all the first part of this talk is really taken from this really good paper, I think a very clear paper by van der Wiel from a few years ago. So here's just the CASP example. So under the additive model, we're going to say that, that if P11 minus P10 minus P01 plus P00, that is actually what happens if you take the this minus this whole quantity. So the additive effect of the joint effect minus the effect of the genetic effect and the environmental effect summed. If that's over 0, we call the interaction positive, and if it's under 0, we call it negative. So in this case, we've got 0.64 minus those two plus 0.3 is positive. So we say that there's a positive interaction, so that the effects of G and E combined are larger than expected based on the sum of the G and E effects. For the multiplicative, it's similar, except we're talking now about risk ratios. And so we get the risk ratio of the joint effect, which is 0.64 over 0.30. And we ask whether that's going to be different than what's the product of these two different E and G effects. And if you do all that out, what that is equal to, the interaction effect is the risk ratio of 1:1 divided by the product of the other two risk ratios, the genetic risk ratio and the environmental risk ratio. So in this case, that's P11 times P00 divided by P01 times P01. In this case, it's 2.06. If that is over 1, we say it's a positive interaction. If it's under 1, we say it's negative. Here we have a positive interaction as well. So we have in the additive scale, a positive interaction. On the multiplicative scale, a positive interaction. But it's important to recognize, and it's talked, and people say it a lot, but that the choice of scale and also the choice of model will dictate often whether the interaction is positive, negative, or doesn't exist at all. So the interactions can be dependent on the scale upon which Y is measured or how Y is modeled. So here's a situation, right, with these particular probabilities in which the additive effect is positive, right? 0.4, the joint effect, or at least 0.4 minus 0.2, the joint effect is greater than what would be expected from the sum of the genetic effect and the additive effect and the environmental effect. But the multiplicative interaction in this case is lower, right? So here, right, you get a 5-fold increase for E, and like a 7-fold increase or something for G. And so 7 times 5 is 35, and that's substantially lower than what you get from what the expected joint is of 20 here for the joint effect. And so in this case, right, this situation, we have a positive additive interaction and a negative multiplicative interaction. Of course, the reverse can also happen, where you have a negative additive interaction, a positive multiplicative interaction. So it's often the case that in real data, that the multiplicative model and the additive model are in the same direction. But there are cases that I've observed, in the literature and otherwise, where they actually do differ in real data. One is positive, the other is negative. And even if they're in the same direction, it's often the case that the p-value for one will be less than 0.05 and the p-value for the other will be greater than 0.05. And so it is important to I think, to discuss to discuss these issues in a paper about whether the interaction is existing in one scale but not the other. So there's a lot of debate about which scale should be used when modeling dichotomous outcomes. So I think this is right. This is something that my grad student pointed out yesterday, Richard Border, that the G by E term in a probate regression corresponds to those in a liability threshold model. But I've almost never seen probate models used in G by E research. What's almost always used is logistic regression. And I think those I haven't done the math on this, but from just looking at it, at different examples of it, I think the logistic regression does give you answers of the G by E that are close to the liability threshold. But they are, in any event, they are multiplicative interactions when you're using logistic regression or log linear models. Those are multiplicative interactions that are being tested. But it's been argued that the additive scale is the one that is of the greatest public health relevance. If what you're interested in is not, ratios, but is total number of individuals that can be saved by treatment, for example, that it's the additive interaction that's actually of greatest public health relevance, because it tells you which subgroup, which level of the moderator for G, for example, is going to benefit most by the environmental intervention, for example. Finally, in this last part of the scale, of talking about scale for this talk, just point out that it's exactly the same thing when Y is continuous. If you're using ordinary least squares and you regress Y on G plus E plus the product of the two, that's testing the additive model. If you're using the log of Y, on the other hand, that's testing the multiplicative model. This really matters, whether you're talking about additive or multiplicative, for non-crossover interactions, because that's just where the order of Y hat changes across for one predictor is reversed across the levels of another moderator. Because there's always, for non-crossover interactions, there's always some transformation of Y that will remove all evidence of the interaction. And so some people call these, interactions that do cross over true interactions, because they're not removable by scale. But when I say not removable, that doesn't mean that the p-value won't become non-significant. So in a sense, all interactions are potentially removable by changes of scale if you're defining an interaction by statistical significance. And just more generally than just talking about transformations on Y, any nonlinear transformation of Y, G, or E can affect the statistical significance of G by E. So it's kind of a tricky situation. So advice with respect to scale issues. So at first, I think we need to stop reifying interactions and talking about whether an interaction exists or doesn't exist, or whether an interaction is real or isn't real. I think that if you observe an interaction on a particular scale of Y, then there is an interaction on that scale of Y, but I think it's a mistake to think that it's real in any kind of real sense, because it will depend often on the scale upon which Y is measured. So, I think if you found a scale of Y that is interpretable and used, and you notice an interaction on that scale, then I think it's fine to discuss, to say that there's an interaction on that scale. But I think it is important to convey to readers that, that interaction may well disappear, if you change the scale of Y. And if we're talking about, whether to use multiplicative or additive models, whether to model it as an ordinary least squares or logistic regression, or, just Y regressed on GE and the combined effects, or the log of Y regressed on those. My feeling, even though it complicates things a little bit, is just to report both sets of results and let readers do with it as they will. I think if you see an interaction on both, then you're certainly in better standing than if you see it only on one or the other scale. So I'll talk now about just how to model G by E and the proper interpretation of terms. This is something that I've done a lot of reviews of G by E, especially in candidate gene literature, and this is something that people get really wrong, even though it's very, it's really very basic. So talk about this for just a sec. So we can model Y as, actually I should have a residual term here. So we can model Y hat as being, okay, I guess I've left off all the hats just for simplicity. So we can model Y as being the intercept plus the effective gene plus the effective E plus the effective.
Can everyone mute, please?
Yeah, there's somebody who's not muted.
Basically, everything depends on everything. You gotta be really careful.
Hello, can you mute, please, whoever's unmuted? Okay, anyway, so GE here is literally the product of the G and the E variables. So beta, I should say beta I here, tests what's called the bilinear interaction. And the interpretation of the interaction beta is that as you increase 1 on E, what is the change in the slope of G? Or you can do it the other way around. As you increase 1 on G, what is the increase or decrease in the slope of E? So that's the interpretation of beta I. And you can see that just pretty simply by rearrangement of terms here. If you take the two terms containing E and you take that out, so this is going to be the predicted Y is the intercept plus the effective G plus a conditional slope of E, which is beta E plus beta I times G. So beta star E is the slope, therefore, when G is 0. It's the slope of E when G is 0, and that slope will increase by beta I for every 1 increase in G. So I think that's what I've said there. So that's what I say here again. So when G is 0, beta star E is just equal to BE. So really, BE, the simple effect in regression, is kind of like, it's kind of analogous to an intercept term. It's the effect of E when G is 0. And that's important because a lot of times people will report simple effects in interaction models when the level of 0 either isn't explained in the paper, in which case you have no understanding of what that actually, what that is actually saying, or even worse, when 0 is completely outside the range of the data, in which case it's completely nonsensical to be interpreting it in the same way it's nonsensical to be interpreting an intercept that's outside of the range. So when interpreting the simple effects in a model, beta G or beta E, that contains a product term, you have to report the coding and you have to be aware of what that actually means. The effect of beta G in this model is the effect of G when E is 0, and the effect of beta E is the effect of E when G is 0. So as an example, say that E is age. In this model, beta G is going to be the extrapolated predicted genetic effect at birth, right? Which may or may not be of any relevance. If you mean center age, right, which is often recommended for these types of models, really for interpretability more than multicollinearity, that's kind of often misunderstood. But if you mean center age, now beta G does have a better interpretation. It's the genetic effect, the predicted genetic effect at average age. Right? So a lot of times people will recommend that you center your G and E variables before creating the product term, and then include all those, the centered variable and the product term in the regression equation. And then these two effects are interpreted as kind of the effect of the genetic effect at the mean environment and the environmental effect at the mean genetic level. So this just goes through, again, just kind of going back to this additive versus multiplicative if we have Y as dichotomous. So in this model, right, this is actually just an ordinary least squares model. So in R, it is literally regressing 0, 1 vector onto G by E. And this is how you write it in R, but really the model is G plus E plus the interaction of those two. That might sound weird that you can use OLS on a dichotomous outcome. But I think that's statistically justifiable once your sample size gets big enough, because even though your residuals are very non-normal, due to the central limit theorem and the law of large numbers, the distribution, the T distribution, and the distribution of betas becomes pretty close to T. And so there's a paper by Hellevick that goes over this. It says, hey, this is making a mountain out of a molehill to say that you always have to use logistic regression, for example, if your outcome is dichotomous. They make the point it's perfectly fine to just use ordinary least squares. Anyway, if we do that, corresponds back to that additive model we were discussing the first part of the talk. And if we do that, the intercept is interpreted as the probability of 0, 0 of getting of the probability of disease when E is 0 and G is 0. Beta G is P10 minus P00. Beta E is P01 minus P00. And beta interaction is the interaction effect of the additive scale. So it's P11 plus P00 minus these two P's, P10 and P01. And I show the derivation of how to get that down there. On the multiplicative scale, we could use a log linear model. And so in R, you would do it the same way, but use GLM, and you say you give the family is equal to Poisson. It doesn't mean it has to be Poisson distributed for Y. It just means that the link function is log. And that changes this to the log of the outcome rather than just the outcome itself. And so if we exponentiate both sides, you get that the probability of Y is equal to 1 given G and E is equal to the product of all these different exponent terms. And if you about calculate, you'll see that the exponent of B0 is again P00, but the exponent of BG, for example, is actually the risk ratio of the genetic effect, P10 over P00. And the exponent of the environmental effect is P01 over P00, or in other words, the risk ratio of the environmental effect. And finally, the exponent of the interaction term is exactly what we'd wanted it to be back before, which is the risk ratio of the joint effect divided by the product of the other two the genetic and environmental risk ratios. And here again, this slide 25 is just for the logistic model. It's the same thing, except you say family is equal to binomial. And beta 1 here still tests the interaction, but it's now on a different scale. Instead of it being a ratio ratio of risk ratios, it's a ratio of odds ratios that the interaction is testing. But they're actually end up being very, very similar unless the prevalence becomes high. So advice with respect to modeling issues, I think first of all, it's important to recognize that the simple effects when the model contains a product term, so beta E and beta G, are predicted effects of E when G is equal to 0, and the predicted effects of G when E is equal to 0, respectively. It's often not gotten correct in these papers. And because of that, I think it's really important to make sure when you're dealing with interactions that you report the scale and the coding of E and G in order to help the interpretation of these simple effects. It turns out that just a linear transformation of G and E does not change the interaction at all. So that the interaction won't change if you center the variables, but the beta E and beta G definitely will change, and it's because the interpretation of them changes. If you center them, these effects are now the predicted effects of E at the mean G and the predicted effect of the gene at the mean environmental level. And finally, for case control data, I haven't seen this done a lot, so I have some qualms about recommending it, but I certainly maybe would be something interesting to look into. But certainly from Hellevik and others, I think it's fine just to use ordinary least squares using a linear model if you want to test the additive model. If you want to test a multiplicative model using a log link function or a logistic, this should actually not say Poisson link function, it should say log link function, log or logistic link function. You can use that in GLM and R. But just do realize that the decision of whether you use, logistic regression or OLS tells it, that really is changing the scale of Y. If you're using OLS, you're testing an additive interaction. If you're using logistic regression, you're testing a multiplicative interaction, and those can be very different. Talk a little bit about proper control of covariates. In genetic epidemiology, we're often concerned about the potential for confounders like ethnicity on a genetic association. And the exact same thing should be of concern in G by E research. So it's, an observed interaction effect, G by E effect, may be due to some unmeasured or measured, confounder. But it turns out that almost always, I think it's like 49 out of 50 cases in which I looked at this, the covariates are modeled incorrectly in such a way that they have absolutely no influence on the interaction term, and therefore the interaction is in no way controlling for those confounders. And in almost every case of those 49 cases I looked at, the authors claimed that they had properly controlled for confounders. And so basically at least in the candidate gene literature, all the candidate gene G by E literature, the G by E term has not properly controlled for confounders at all. So, if you imagine what people would say if an entire branch, right, all candidate gene studies didn't control for anything, that's pretty much where we are for G by E studies. They really haven't controlled for anything at all. Just kind of walk through how this works. So if we let C be the variable we want to control for, and here I'm just going to talk about a single covariate, but obviously this generalizes to multiple covariates. So C, it might be ancestry, the first principal component from an ancestry GRM, first ancestry PC from a principal component of a GRM. So the incorrect but usual way to control for C is just to include it, the main effect of it in the model. But by doing this, this actually has absolutely no influence on the interaction term, and therefore it doesn't control for the potential of that confounder to influence the interaction term. Actually, the correct way to model this is to include the covariate by G and the covariate by E interaction terms in the same model in which you're including the main effects and the covariate. And if you do this, the effects of the covariate on the interaction are not properly controlled for. And I should note that this applies to any kind of test, any kind of G by E test, and equally therefore to what are called GWIS, genome-wide interaction studies. So, the C by SNP and the C by E interaction terms have to be included at each SNP in order to properly control for this. And otherwise, you can get really serious inflation of test statistics to the degree that those covariates are responsible for the interaction. So let me kind of walk through why this works. So if we say that C is the covariate, the dummy codes just African ancestry versus European ancestry, and we have a mixed ancestry sample. Obviously, this is not going to happen often, but this does happen often if we're talking about a European-wide sample, and C would be the first principal component that differentiates Northern versus Southern European, for example. So this, these things do happen. So we're interested in whether the outcome is Alzheimer's disease is affected by ApoE, which is G, and carbohydrate intake and their interaction. So does the effect of ApoE depend on carb intake on Alzheimer's? So if it's the case that the E4 variant, this is actually true, is more frequent in people of African descent, and if the effects of carb intake on Alzheimer's disease is different for Africans than it is for people of European descent. So the effect of, there is actually then a carb by ancestry interaction, right? Then the apparent G by E interaction that you observe may just be an ancestry by E effect. And that's not going to be controlled just by entering in ancestry as a main effect. And it can work the other way, right? If people of African descent, say, eat more carbs or fewer carbs, whatever, there's a difference in carb intake between African individuals of African descent and European descent, and the effect of E4 is different between those, or the effect of E4 and AD risk different between African and Europeans, then an apparent G by E effect may actually just be a G by ancestry effect. And both of those scenarios are appropriately controlled for here. So in the next slide, the next two slides, I give a couple of, kind of examples of that. Oh, hold on just a second. Okay, let me just, okay. Okay, so this is the first example I talked about. So here we've got, let's say that, that we have this carb by AD risk interaction. So there is no ApoE by carb interaction. What's actually is going on is there's an ancestry by carb interaction, so that African Americans carb intake matters a lot to Alzheimer's disease risk, and for Europeans it doesn't really matter at all, maybe due to background genetic effects or something like that. If it's the case that there's an ApoE difference between African and Europeans, which there is, this is going to lead to increased evidence for an ApoE by carb intake interaction, right? So here, this would be the E4 allele, and here we'd have the E3 allele, and it looks like the effect of E4 depends, the effect of the polymorphism depends on whether you have low carb intake or high carb intake. And that's obviously not going to be controlled for if we just, and the reason this is if we just include the main effects. The reason this is because, E4 is really a weak proxy for ancestry, and E3 is a weak proxy for being European. And therefore the true ancestry by E effect is masquerading as an ApoE or a gene by environment interaction. And that will only be controlled for if we include the ancestry by E interaction term in the model. And this is the same thing here. I mean, here we've got, you know, ApoE, and if it's the case that, you know, ApoE is E4 is really bad for European descent people, but E4 matters less for people of African descent. And this is actually also seems to be true. If there's a difference in the amount of carbs consumed, this is going to come out looking like an ApoE by gene interaction. And obviously it's, you know, an issue of power of whether it will be significant or not. And, you know, I'm making this out to be false interactions. It also can be the case that you can have a true interaction that is masked by these covariate effects that you will therefore commit a type 2 error. You won't observe a true interaction unless you properly control for things. And that just describes that here. I think this applies to polygenic risk score models too. So the correct way to use the covariance in a polygenic risk score study is the you know, the main effect of the PRS plus the environment plus the interaction between PRS and environment, plus the covariate terms. And here, if you just have a single covariate, then it would just be the main effect of the covariate plus the covariate times the PRS plus the covariate times E. And again, I think this is routinely being done wrong in the PRS literature. So there's this paper by Luthia Kolodro-Conde, who he's a good friend of mine, and a lot of other people who are good friends of mine on that paper, many of which you know a lot more about statistical genetics than I do. So this, I'm not trying to like one up or anything like that here, but I think that they've gotten this wrong. And in fact, they cite my paper of how to do it correctly, but then they proceed not to, they proceed only to control for the simple effects in that paper. And this just, you know, again, this maybe is getting a little bit redundant, but if you've got, you know, so for example, MDD PRS from low to high, and we differentiate by, say, Northern Europeans versus Southern Europeans, it might be the case that the initial sample from which the beta weights were, you know, regardless of whether they control for PCs in that initial sample, the beta weights are more strongly weighted towards those of Northern European descent, because the sample composition, for example, then it is going to be the case that the PRS is going to predict MDD better among individuals of Northern European descent than among Southern European descent. And all it takes would be something like some mean, you know, mean difference in stress and life event stress between Northern and individuals of Northern European descent and Southern European descent in order to induce, you know, a false interaction that looks like that you're getting a stress by MDD interaction, when in actuality, what happened is that you didn't properly control for the ancestry covariate in this case. You know, so that's kind of what I say here. So advice with respect to covariates, you know, I think we wouldn't dream of running a GWAS without properly controlling for covariates. And I think interaction studies demand the same level of rigor. Otherwise, your type 1 and potentially type 2 errors can go through the roof. For candidate gene, GWiz, and PRS studies, I think we really need to start properly controlling for covariates, or at the very least stop claiming that we've done so. Including main effects of covariates does nothing to control for the potential covariates influence on interactions. And this is this last thing is about, you know, well, what about GREML and G by E analysis? I'm not, to be honest, I'm not totally sure, but I think, I mean, at least you know, it kind of seems parsimonious to think it's going to be the same kind of issue, that you need to control for a G by covariate random effect as well as an E by covariate fixed effect. Obviously, when you start controlling for all these effects, if you've got 10 ancestry covariates and gender and income or whatever, you're talking about, you know, 12 covariate main effects plus 12 covariate interactions with E plus 12 covariate interactions with G. You know, you are going to start whittling away potentially at the effect, but that is the whittling away should happen, right? Because that is shared variance in which it's unclear what's actually going on. And that's, and then certainly, you know, hopefully we have a big enough samples and enough degrees of freedom that controlling for an extra 30 or so covariates shouldn't be catastrophic to our power. Okay, so I've actually done great on time. I'm impressed with that I got this far. But anyway, so recommendations. Final slide is just so you know how do I think we should do G by E analyses with respect to at least some of the issues I discussed here. And I'll say one other thing at the end of the slide, and then I'll open it up to questions if people have. So first, you know, I think that it's important to recognize that interaction studies are hard to do. And they're open to alternative and equally valid interpretations due to differences in scale or differences in how they're modeled. And also that interaction studies are underpowered the tests of interaction effects are underpowered compared to those of main effect tests. So I think first you just, before you jump into interaction research, think about whether it's worth doing it, because, I think it might be easy to go away from a talk like this or papers that are written about this kind of stuff and say, well, that's just a bunch of complicated statistical mumbo jumbo and I'm going to ignore it. But I think that if you're going to do interaction studies, it's really incumbent, I think, on you, the investigator, to do it properly. And it's not, it's not scientifically justifiable just to say, oh, it's confusing and therefore I'm going to ignore it. No, I think if you're going to do it, you have to embrace that it's hard and try to do it properly. So, I think, so there's a lot of candidate gene by environment interaction studies. I've been a critic of those, so I think if in choosing to do that, I would try to, probably preaching to the choir here with this audience, but probably stay away from trying to choose particular candidates rather than that, maybe look at the aggregate effects of SNPs using GRML or PRS, or maybe doing it across all the SNPs using a GWiz. I think it's important that we, directly report the additive and at least the additive and the multiplicative scales of Y when we're doing this, the additive multiplicative models, because the effects may well be very well may be different between those two, and I think that's information that's important to convey to the reader. It's important to center. Whoops, I don't know why that happened. To center your let's see where I'm at. To center the G and E variables, and that helps in the interpretation of the simple effects in the models. And if you don't do that, then at the very least, it's crucial to describe what the zero level means of G or what the zero level means of E, in order so that readers can interpret the simple effects. I've seen a lot in these papers where an interaction term is included, and people will then start interpreting the simple effect of G or the simple effect of E and saying, oh, it's very interesting now that after we've controlled for the interaction, there's now a big effect of G. Well, that actually makes no sense. And probably what's going on is that the zero level of E is outside the range of the data. And now you're talking about some extrapolated genetic effect among people that don't exist in the real world. And finally, it's important to properly control for covariates by including their interactions. So including the covariate by E and the covariate by G terms. And similarly with PRSs, it's important to include the C by E and the C by PRS terms. And for GRML, I think this still requires more work. Okay, so thank you for being with me, and I'm happy to take any questions that people might have. I think I'm going to push stop share here. There we go.
Okay, thank you, Matt. That was fantastic. So I think there's a lot to think there. Has anyone got questions? I've got lots if no one wants to speak up, but unmute yourself, Jerome. I've seen you unmuted.
Yeah, so I've got a naive question. So encoding G and E as 0 or 1,
yeah.
Are we ignoring the fact that you might have protective genetics and protective environment, and it should be -1 to +1 in both cases?
I think, I think it's kind of arbitrary. I think if you want to do G as -1 and +1, that's fine. It just changes what the interpretation of the main effect of E is. The simple effect, I should say main effect, I should say simple effect. The simple effect of E in that situation is going to be the predicted effect of kind of the mean between 0 and 1. So it's not necessarily the predicted effect at the mean, it's the predicted effect of the mean of the two means.
I'm thinking that this would be more of a problem for oligogenic disorders or for environments that are toxic are not, right? So that's 0, 1. But environments like parenting, for example, where it can be very good or very bad.
Yeah.
So I mean, that's a different.
Right. I mean, I think it's just up to the researcher about how they want to code it in a way that maximizes the interpretability of the simple effect terms. I think what's crucial is that you just describe to the reader how it's been coded so that the simple effect terms can be properly interpreted. What is totally wrong, though, is to not describe the coding and then to start interpreting those simple effect terms as if they're not conditional on what the coding is of the other variable. The effects depend on the 0 level of the other variable, so it's therefore crucial to explain what that is.
Anybody else?
I was just going to interject there that it's important that the coding won't affect the significance of the actual interaction term at all.
That's right.
So from that perspective, it doesn't matter.
That's right. So really this issue of coding is just about interpretation of the simple effects. And, it's been argued, I've seen it argued that people have said, look, don't even interpret simple effects in interaction models. Interpret them in models that don't include the product term if you want to interpret the simple effects. And then if you want to interpret the interaction, include the interaction, but don't interpret the simple effects. So, I mean, that's another strategy, and that's perfectly fine. It's pretty close to being the same as centering the variables. But in any case, what's important here is interpretation of the simple effects, and people just get that really wrong sometimes. But as Richard, that's my graduate student who's doing some of this interaction research with me, said, it does a linear transformation of GxE will not change the significance of the interaction term at all.
Paul, I think you've unmuted.
So on the point of controlling for covariates, I remember thinking about this once and coming to the conclusion that it only mattered for confounders. And the examples that you gave were all ancestry, factors. Yeah. Does it matter for factors such as gender and age, which are not associated with genetics and therefore not confounded genetics?
That's a great question, actually. So it won't matter if there's no association at all between genes and what other variable that you're interested in.
It's just population structure then.
Yeah, I mean, I think though, if you're going to control for mostly just probably population structure, but if you're going to control or claim that you control for gender, right, then I think then you should go ahead and control for, I mean, if you're going to do that in the main effect model, in which case we know there's usually no effect of gender on the frequencies of alleles, although there's an exception obviously with the X chromosome, and depending on how you've modeled that. But I think if you're going to claim that you control for gender in an interaction model, I think you should just control for it properly in the same way that you can, you control for it in a main effect model, even though we don't expect there to be any little frequency differences between gender. There is a situation, right, with where this becomes crucial in modeling the polymorphisms on the X chromosome, because people will model, a 0-1-2 or 0-1 for females if you have the risk allele, and 0-1 for male if you have the risk allele. And there, due to Hardy-Weinberg, the low frequency differences are actually quite different between the genders. And so it becomes crucial that you model it correctly. And this was done incorrectly for some of the candidate gene MAOA research. And actually, if you read those results, it actually makes a lot of sense what they found with respect to not properly controlling for confounders.
Okay, but in a sort of main effects standard GWAS, you would only expect to get type 1 error if you didn't control for population structure.
Right.
So what exactly are you saying here? Because in a lot of cases, it will make a big difference whether you only control for PCs or whether you need to control for all interactions with every possible covariate that you would have put into a Unifex model.
Right. I think that I think if you're doing a GWiz in which you're looking at the interaction effects, right?
Yeah.
So you're looking at the SNP by stress interaction across the genome, right? Sure. For each individual SNP. I think it's absolutely crucial that you've controlled for the SNP by ancestry and the ancestry by stress interaction in every one of those models, right? And then if that's 10 PCs you're controlling for, you've got to have 10 ancestry by stress interaction terms included, and so yeah. So I think that's absolutely crucial. I don't know. I haven't seen if people are doing that in GWiz studies. I haven't seen a lot of GWiz studies out there actually, but my concern is that. Go ahead.
Oh, I was just going to say it's pretty uniformly not done. I've never seen it done.
So you've seen GWiz studies and they don't do it?
Yeah, exactly.
Okay.
Okay, I guess it would just be good to differentiate between covariates and confounders.
Well, I mean, the usual reason we include covariates. Well, okay. So you're saying that you might include a covariate not because you're worried about it confounding the effect.
Yeah, to increase power.
Clean up. Yeah, you want to clean up the. Right.
And
well, that's an interesting.
There'll be a big difference in the number of predictors that you end up including if you feel that you need to include every interaction.
Yep. Well, okay, but I mean, first of all, we usually have plenty of degrees of freedom in these models, so that shouldn't affect our power too much. And if part of the motivation is to, clean up variance in Y, is to remove some error in Y, some error variance in Y, then including the interactions would do the same basic thing, right, as including the main effects. If there's interaction effects that are of no interest that we want to remove, then I'd say that, the motivation for including it in the main effect model should be the same as including it in the interaction model. So even if you don't worry about it being a confounder per se, I could see the argument for doing it. Although I think you've got a good point. I think there are situations where including these covariates just it's not crucial for proper, for controlling for confounders.
Yeah, I just wonder as well with logistic regression whether computational time could be an issue. I'll take your point about degrees of freedom. If you've got big enough data, then it's fine. But
yeah, right. Yeah, possibly. But again, it's one of these issues of it's like, well, if that's how it has to be done, that's how it has to be done. I mean, if just because it takes longer, it's not an excuse for not doing it correctly.
Sure. Yeah.
Any other questions? So Matt, one of your conclusions was that you should always report additive and multiplicative results, but could you not say that, would other advice be that at the beginning of your analysis, you should say, this is the reason why I'm interested in doing a G by E analysis. This is the scale that I'm interested in the result on, and then make that your primary result, so that you're directly stating why you're doing the analysis and what the scale that's of relevance. Right. And then maybe secondarily you can say, I recognize that if I do analysis on a different scale, the interaction goes away, but the point is that on a scale that's important in public health, then this is the scale where I see the interaction.